All right, let's talk about the most critical, and honestly, the most frustrating part of any Western blot, your sample prep. It is, without a doubt, the first step, and it's the one that decides if you get that beautiful, clean data or, a complete and total waste of your time. Come on, we have all been there, right? You spend days on this thing. You follow every single step perfectly. You run the gel, you do the transfer, you're incubating for hours. And then what happens? You're met with that soul-crushing sight of a completely, utterly blank film. It is one of the worst feelings in the lab, for sure. So you ask yourself, where did it all go wrong? Well, more often than not, the whole mission fails before it even gets off the ground. Your entire Western blot, everything you do downstream, hinges on that initial protein extraction. If you mess that up, nothing you do later is going to save it. Okay, so let's think about this a different way. This isn't just a boring lab protocol. This is a high-stakes operation. Let's call it the Great Protein Heist. Your protein of interest, that's the prize, and it's locked away inside this incredibly complex cellular vault. And your lysis buffer, that's your specialized toolkit for cracking the safe. Every good heist starts with a solid plan, right? And for us, that means doing some serious reconnaissance. Before we even think about breaking in, we have to know exactly where our target is hiding inside that cellular vault. So, is your protein just kind of floating around in the cytoplasm? Or is it really stuck in there, embedded in a membrane? Maybe it's locked down deep inside the nucleus. See, each of these locations is like a different room in the vault, each with its own security system, and each one needs a totally unique approach to get in. And this right here, this cheat sheet, is basically your strategic blueprint. It maps the target's location to the right set of tools. So if your protein is out in the open, in the cytoplasm, a milder buffer like NP40 will probably do the trick. But for those targets that are hidden away in the nucleus or mitochondria, you gotta bring out the heavy-duty equipment. That's gonna be your RIPA buffer. Why? Because its powerful detergents are literally designed to blast through those tougher, more complex membranes. Making this choice is absolutely mission critical. So we've cased the joint, we've identified the target's location. Now it's time to actually open up our kit and choose the specific tools we need to get the job done. We're talking about the lysis buffer itself. So what exactly is this tool? Well, at its core, a lysis buffer is basically a chemical cocktail. And its most important ingredients are these things called detergents. You can think of them as molecular crowbars. They are designed to pry open the cell's membranes and get your target protein out into the solution where you can work with it. And this really illustrates the difference in your toolkit perfectly. A mild buffer, like NP40, it uses these gentle, non-ionic detergents. It's like a precision lockpick. It's perfect for those easily accessible cytoplasmic proteins. And it's really crucial when you need to keep protein-protein interactions intact. But on the other hand, you've got a harsh buffer, like RIPA. It contains aggressive ionic detergents like SDS. This is your explosive charge. It's going to blast through everything to release those really tough, hard-to-get proteins from deep inside the nucleus or mitochondria. Okay, you've picked your tool, you've breached the outer walls of the cell. But here's where the heist gets really clever. The second you crack that safe, you're going to trigger all the internal alarms. So we need a plan to disable the cell's security systems before they destroy our prize. You have to understand that the second that cell membrane breaks, all hell breaks loose inside. The cell releases its own defense mechanisms, a flood of enzymes called proteases and phosphatases. Their only job is to chop up and deactivate proteins, and believe me, they will go after your target immediately. The clock is officially ticking. And this is where your countermeasures come in. You have to add inhibitors to your lysis buffer. Think of these as specialized little gadgets that you use to disable specific alarms. PMSF and cocktails with things like lupeptin, they jam up the protein-shredding proteases. EDTA and EGTA, they basically cut the power to a different type of protease by grabbing the metal ions they need to work. And hey, if you're studying phosphorylation, sodium orthovinidate is non-negotiable. It's going to block the phosphatases that would literally erase those precious signals you're looking for. And here's the final briefing. 3 simple but absolutely non-negotiable rules for this heist. Rule number 1, keep everything on ice. Cold is your best friend. It slows down all those destructive enzymes. Rule 2, pre-cool all your tools, your buffers, your tubes, the centrifuge, everything. And rule 3, add your inhibitors fresh, right before you use the buffer. They degrade over time, so doing this makes sure they're at maximum strength, right when you need them most. Okay, the plan is set. Our tools are selected. Security is about to be disabled. It is time. Let's execute this heist step by step. So it all breaks down into 5 key actions. First, you're going to prep and wash. Clean the scene by washing your cells with ice-cold PBS to get rid of any contaminants. Second, you lyse. Add your chilled lysis buffer with those fresh inhibitors to crack open the vault. Third, agitate. You gotta vortex or sonicate to make sure every last cell is broken open and to shred any of that sticky DNA that gets in your way. Fourth, centrifuge. Spin it down hard. This separates the prize from the rubble, pelleting all the insoluble junk to the bottom. And finally, step 5, collect. You carefully take that clear liquid on top, the supernatant. That right there is your treasure. That's the protein lysate. You've made it out, you have the goods, but the mission isn't over yet. Look, a professional doesn't just grab the bag and run. They confirm the score and they prepare for a clean getaway. You can't just run with the bag, right? You need to know exactly what you've got, and this is the protein quantification step. It is absolutely essential if you want to compare your samples accurately in the final analysis. So, here's the final prep for that clean getaway. First, you quantify your protein concentration. You'll probably use a colorimetric assay like BCA or Bradford. Second, and this is so critical for getting reliable data, you normalize them. This means you do a little math to calculate the exact volume of each lysate to load so that every single lane on your gel has the exact same amount of total protein. And finally, you prep for loading. You mix your normalized lysate with Lamley's sample buffer and you heat it up. This denatures the proteins, basically getting them all stretched out and ready for the final stage, running the gel. Ultimately, it all comes down to this. The story that your final Western blot tells, whether it's a huge breakthrough or just a total bust, that story is written in these very first steps. The entire experiment rests on the quality of your protein heist. So the next time you're at the bench, ask yourself, are you just mixing reagents or are you actually planning for success?